Hello, everyone. Thank you for joining our webinar, Microbiome Analysis for Probiotics, Considerations and Challenges. This webinar will be hosted by Megan Stike of Eurofins, and our panelists include Dr. Greg Siragusa of Eurofins and Dr. Thomas Tompkins of Lallemand Health Solutions. I'm Genevieve Randall, and I'll be moderating this webinar. Before we begin, I'll let you know more about how this webinar will run. The webinar is being recorded, and the slides and recording will be available for you within 3 business days. A short Q&A session will follow the presentation to answer viewer-submitted questions. We will also be pausing halfway through just to answer any questions at that point as well. During the webinar, you can submit questions you have using the webinar sidebar menu. Select the Questions tab, type in your question, then hit the Enter key on your keyboard. Remember, you can submit questions throughout the webinar. Now I'll give a quick overview of Eurofins. Eurofins is driven by our mission to contribute to global health by offering the highest quality testing, training, auditing, and consulting services. We strive to listen to our customers and not simply meet but exceed their expectations. Our footprint is global, with 35,000 staff and 400 laboratories across 44 countries, and a portfolio of over 150,000 analytical methods. Eurofins provides a unique range of analytical testing services to the pharmaceutical, food, environmental, and consumer products industries and to governments. Okay, before our speakers begin their presentations, we will have a quick polling question. And the polling question is, have you utilized microbiota analysis in the past? Yes or no? So you may click on the answer on your screen, and in about 30 seconds, I will close the results and share them with everyone anonymously.
Thank you, Genevieve. That's, it looks like we've got a good percentage of attendees that have not used microbiota analyses. I believe this presentation will help many of those who have and have not. So starting out with a few questions, microbiomes are now a reliable and standard tool in many areas of microbiology and in clinical trials and finished product testing. Dr. Siragusa, could you start out with describing and defining what a microbiome is?
Yes, thank you, Megan. If I could have the first slide. A microbiome is the descriptor for a population or community of microorganisms, be they commensal, symbiotic, probiotic, starter culture, or even pathogens that share a space. A microbiome can be bacterial, which is what we're really going to focus on today. But a microbiome can also be a microbiome that includes fungi, viruses, yeast, bacteriophage, and specific genes, such as antibiotic resistance genes. The process to obtain a microbiome on the bottom right is pretty straightforward as a process, but we'll see that there are a number of idiosyncrasies to discuss. We start with a sample, we extract that DNA, we amplify our target, we sequence it, and then we do bioinformatics, the fun part, and in a lot of ways, one of the more difficult parts. And we wind up with an output of, in this case, represented by a pie chart of different bacteria in a sample. If I could have the next slide. Microbiomes as a tool have revolutionized many parts of microbiology. Previously, relying strictly on culture, we are not able to obtain identification to a genus, species, or subspecies level without um, long and rather arduous methodologies that take a lot of time and have varying degrees of accuracy. Microbiomes are based upon a sequence, which can be a discrete, reproducible unit that's portable to all labs across the world. And databases will bring us to the genus, which is in the upper box, or more specifically, depending upon the size of the sequence, can bring us to the strain level. And we see here as an example several strains of one organism, Lactobacillus plantarum. That's the power and the uniqueness of microbiomes.
To me, this is Thomas here. To me, the microbiome has become the new 42. I'm an end user. My interest, and what I mean by 42, is it's the answer to the life, universe, and everything. If you're a Hitchhiker Guide to the Galaxy fan, you'll understand the reference. So it becomes, and it has become, the application is to understand how the microbial composition or the functionality of the microbiome can now be used to either predict disease or to predict, hopefully, healthiness and how we can restore healthiness through intervention, through probiotic intervention, through prebiotic intervention, or other interventions. It is a double-edged sword, though. There is this opportunity that I've talked about. There's the potential to understand deeper and make a potential correlation, but not necessarily causation between the evidence you receive and the outcome, the clinical outcome that you're looking for. But again, it could be misinformation. You'll see a lot of information out there that is misleading because the work has not been done in an appropriate fashion, or they're making trying to make a causation out of a correlation. So we need to look at that. And I would say, unlike Megan's title, I would say this is probably not reliable at all at this time. I would argue that we're still at the Wild West stage, and there's a lot of things that we need to really dig into, and hopefully that's what we'll talk about today, the challenges and considerations before we really learn how to harness this and how we can use this effectively in our industry. So thank you very much. Next slide, please.
Thank you, Dr. Tompkins. Certainly, microbiota analyses can be the Wild West. I'd be curious, how are each of you and your clients using microbiomes today? Dr. Siragusa, if you would start.
Well, to follow up on the Wild West theme, as a microbiology cowboy, most of our clients today who are in the business of producing probiotics and also in the business of determining how they affect animal models have two main questions they ask at first. And that is, on a product, what is the viable cell count? And if it's a single strain, that can be done culturally. Secondly, is it my strain? Is what I put into the blend actually what was blended? And do I have information that I could use to support a label? Do I have information that would also tell me approximate proportions? How consistent batch mixing is? In the efficacy side of it, microbiomes are now useful for fecal analysis and analysis of other bodily fluids and tissues. These are things that previously were done strictly by culture, which is good in its own right, again, specifically for single strains. But when we have a mixture of strains, as in the intestinal tract or in a mixed probiotic, microbiomes now offer that solution. So if I could have the next slide. This is an example of a microbiome conducted on a mixed probiotic product. And on the left, or the middle column of the table, we see the relative proportions. So microbiomes are not quantitative. Microbiomes provide relative proportional abundance. So it's something to keep in mind. Sometimes there's a misconception that microbiomes will tell you a number of organisms in a sample, and that's not the case. We see that there's a list of organisms, and if you compare that microbiome analysis to what's in the product label, You see there's not a perfect match of correlation, but we were able to identify all the organisms that the manufacturer knew was in the mix and was on the label. And in some cases, we came pretty close. And I think this is a good point to say that microbiome data is not necessarily precise, but it does give us a good snapshot of what's there. And if we look at it again in the graphic form, that's what's on the right.
One thing I would add here, Greg, perhaps, is that, of course, at this level, you still can't distinguish between two similar species, for example. So KCI versus periKCI or rhamnosus, they kind of still group together at this point. So you might need other techniques, which we can discuss later, to get that better resolution.
Yeah, good point. And for the audience, that resolution can depend upon the size of the sequence that you obtain and whether or not you go beyond just 16S genes. The 16S gene is a marker for bacteria. It's not found in eukaryotic organisms in general. Something else I think I should point out, if you look at the bottom two organisms, Anoxibacillus and Geobacillus, those are bacteria that are very common in environmental samples on the planet. They can be found in all sorts of environments, but the way they get into a probiotic mix is that if there is any other adjunct or binding agent or color that is part of that formula, they very well could be there. They're considered to be innocuous, and I put them there because they were present at a very low level.
Greg, and I think this is an important aspect. If you're going to do microbiome analysis on your product, then you must take every individual component of that product and run it through the system, the sequencing system. Otherwise, you wouldn't know necessarily where the source is coming from, and it could be just a contaminant of the system itself. As we know, there are errors that can occur in certain sequencing systems.
A good point, Tom. And you're speaking every component. You mean all the quote raw ingredients that go into the final product. Okay.
Exactly. Every component that's there, you should do analysis on that as well, just to see if there are any background levels of some of these, I would say, water-based organisms and whatnot that you can see coming up on occasion.
So if we can have the next slide. This is another example that one of our clients is using the microbiome to compare effects. And I would start out by saying, and repeating what Dr. Tompkins said, correlation is not necessarily causation. So what we're seeing here are snapshots. These are microbiome snapshots. And these are fecal specimens, and this is actually from an avian model. And they were comparing feed treatments amongst populations and the control group. And what we see, just looking at the microbiomes represented as bar chart, they're quite similar. Now, also I want to add at this time, for anyone who is colorblind, it's probably a good idea to make that known to your provider so they can take steps to help you interpret different colors and shades. If you look to the right, it's the same data plotted three-dimensionally, and we see that there's not really any difference. There's not any real discrete grouping. They all tend to group together. Now, you could say on the right, there are two birds out of the ordinary, the blue and the orange, that group together. Possible, but given the number of samples that were done, probably very unlikely that those are true outliers. But nonetheless, a PCA plot can be obtained from the same data, and you can look for effects to see if you have managed in any way to possibly change the microbiome by some additive, be it a probiotic or a botanical, for instance.
One thing I would mention here too, Greg, you have to be a little bit careful in the interpretation also, because here in this situation, we're mixing both the genus level, the family level, and the order level, and the class level. So we're kind of reporting all of them at the same time. You probably should try to take that down to the same level, whether the genre level or the family level, to make these comparisons. When you start doing these blends, it becomes much more complicated, and you don't necessarily get the answer you should see, but you get the answer you expect to see, put it that way. So you have to be a little bit careful about mixing the classes and the family and the order and the genus level as well.
Dr. Tompkins, I think you raised a good point, and Dr. Siragusa as well, in terms of understanding and asking the right questions at the start prior to analyses. So whether it's looking at your individual raw components and then carrying that through the finished, understanding what level of resolution you need, and also understanding what type of format, table, graph, etc., that you'd like your data to be most useful to the end user, all seem to be important. So Dr. Tompkins, could you walk through how your clients are using microbiomes today?
Absolutely. Thank you very much. Next slide, please. Yes. So we are, we are a probiotic producer, but our clients generally rely on us to run the clinical studies. We have the expertise, the experience. We have all the methodologies in place. We do everything from A to Z. We design the studies, set up the studies, all aspects as post-study analysis too. We do in-house. We bring the samples back in. We do immune markers. We do microbiome analysis. And at every given time, we have about 20 to 25 clinical studies underway either with clinical research organizations, university professors, or medical doctors. And we have a lot of visiting professors who use our know-how, our pipelines that we've developed to aid them in their studies as well. So we actually do a lot of partnering and collaboration with groups who want to learn how to do this. Next slide, please. Now, the questions can be quite simple. Most clients and investigators come to us with a very simple question. Does the microbiome composition change when they add probiotics, prebiotics, or antibiotics to their system? That's sort of where the baseline for where everybody wants to start. Is there something that we can see? Then, once you start digging into it, then you start asking those really exciting questions, the more pertinent questions. How do we find the probiotic amongst all these other microbes? Are there specific changes in certain genre that we can correlate with the outcome, or is there shifts that we see that maybe we need to understand in terms of functionality? So all these questions are actually quite exciting, and this is where you actually begin to make great inroads into your studies and really begin to think about the future. And that's the challenge for us today. Next slide, please.
That's a perfect transition into, we've talked about what the definition is, how our clients are using microbiomes today. So let's talk about what the considerations and challenges are of microbiome analysis that we need to know going into this. Dr. Siragusa, what do you see as the considerations and challenges?
So we could have an entire webinar just on this very topic. What I'll try to do is to give some of the highlights of what you should ask and know to consider when you're designing an experiment with your provider, with your microbiome provider. The process of obtaining a microbiome is pretty consistent across sample types. But on this slide, what is probably the two most difficult things to do or to answer initially is the extraction method and the bioinformatics pipeline. If you move to the next slide, please. So I want to comment about sample types, and of course that will affect which extraction method is used. Dr. Tompkins is going to present some real data obtained from his laboratories on extraction comparisons. But what I want to say is whether you have a dry product, low water activity product, a liquid product, or a semi-moist sample, whether that's from one of your trials, if it's fecals, or if it's actually a food. There's some guidelines that you should know about. That's the quantity, of how to treat it, keep it dry, ship it overnight. If you're sending an agar plate with isolates or even mixed, I'll just say this: don't freeze. Biosecure packaging. That's obvious to many people, but not everyone. I think it's worth noting. Go to the next slide, please.
Yes. So Greg has brought up a very important point here. So much of the success of a clinical study, at least from our perspective, is based on how that sample is collected. So this is extremely critical. First you need to find the best way to get the samples from the participant to the laboratory and get them there as fast as possible. we use taxis, we use students with cars. We go out and get those samples as soon as they're produced and bring them back to the facility to start processing right away. We also use barcoding systems. This is fantastic for minimizing personal errors. Everything from the collection kit through to all the extraction tubes are all barcoded, and this allows us to follow a sample from point A through to the point of final sample analysis very easily. We know how many times it has gone through the freeze-thaw cycle, if it has been frozen. We have access with freezers with card control access, so we know who accessed it, how long they accessed it for, what were they doing with that sample. I think this is a very important aspect as well. And then finally, there's other things that you need to consider. How are you going to get those samples to you? If I'm doing a study in the United States, I'm in Canada, and it's not too bad of a problem. But if I'm trying to get something from Asia, that can be a difficulty. How do I get it here quickly? Is DNA better than fecal? Sometimes shipping fecal is actually easier than shipping DNA. So spend your time preparing the standard operating protocols for these steps. This can save you a lot of problems further down the road. So this is, I think, a critical point. Make sure that you design into your clinical studies the best management of the samples. If you can't do that, then you're going to be lost right from the beginning. Next slide, please.
I think, Dr. Tompkins, if I could just add to that and ask a question of you and Dr. Siragusa. So there's a lot of kits, commercial kits on the market in terms of shipping and assisting in long-term storage or overnight shipping. What are your opinions on those standard kits? Where do those fit into each of your respective SOPs from Eurofins or Lallemont Health Solutions?
Greg, would you like to start us off?
Sure. Just to make a general overview of some of the other considerations, I want to talk about some terminologies. You'll often hear the word pipeline, and for many people, pipeline refers to the bioinformatic steps that are required to go from a sequencing machine to an output that's usable, a high quality output. That is, the different peaks where all of the nucleotides are well separated, taking that and editing away any less than good quality sequences, and winding up with a list of species or genera or orders of, in this case, bacteria. How you get to that point is the pipeline. If I could have the next slide, please. We talked a little bit about databases earlier as being one of the critical points probably after sampling, because in many senses of the word, sequencing has become a commodity, a pretty standardized commodity, probably analogous to printing. That chemistry is on good, solid footing. But databases, let's talk about that. These are three publicly available databases: the NCBI, also known as GenBank, the Ribosomal Database Project, or RDP, and Easy BioCloud. And all of these are databases that have bacterial sequences deposited by individuals, but not all of them are what we call curated. So for instance, NCBI or GenBank is not a curated database. So someone can submit high quality sequences or they can submit low quality sequences, but they're both deposited. In the case of RDP and Easy BioCloud, those are what we call curated. Every sequence that's entered into those databases has been checked for quality. And if we just glance real quickly, you see that the GenBank, in this case, and this is on the same sequence, brought us to the genus level, Acinetobacter. The RDP data brought us to the genus Acinetobacter, but then it describes other strains and other similar sequences below that genus level. And then thirdly, the Easy BioCloud does a similar thing. But what I want to point out here is if we look at the the identity values, you'll see 100%, 100%, 100%, 99, and 99. These are the top 5. And what I want to draw your attention to is the top three were 100%, yet the answers, if you will, are slightly different. And that's due to the database composition. So we have to ask the provider, what databases are you going to use in your bioinformatic pipeline? Next slide, please.
And I think just to add to this, that's a really important piece. As it relates to human probiotics, there are some new taxonomic denotions that are coming out in 2019, expected within this next 6 months. So as you do get your data back, and depending on how you're going to use microbiome analysis, do press back and discuss that taxonomic assessment, assignment in your final report. Sorry, Dr. Siragusa.
Oh, thank you, Megan. And sequencing facilities and choosing a sequencing facility. I just want to make the statement again. If I can drive it home, you should be a partner with your provider. This is a project where you're each contributing a part, and you need to rely on the expertise of that provider. You have to be sure of your objective. If it's the simple question of, is there a difference in these two treatment groups due to a probiotic? You better examine that question and ask, well, do you expect any differences? Are we looking at the right population? Do we need to look at fungi instead of bacteria? You should discuss with your provider the primer sets. That's what it's targeting. You should discuss whether or not you need a targeted microbiome, which looks just at the population of 16 S genes for bacteria and ITS genes for fungi or yeast, or shotgun metagenomes, which is a longer process and at the moment slightly more expensive, but it sequences all DNA in that sample, and it gives you a lot more information to draw from. You should certainly ask about standards, and Dr. Tompkins is going to show us some really interesting data. And if you have knowledge within your group of how to, for instance, lyse the organisms that you produce, you should share that organism with your provider, because that'll spare them a lot of time in choosing the right extraction kit. About the facility itself, you should ask about turnaround times, expected turnaround times. You should ask about contingencies. What happens if sequencing fails? Or what happens if the sequence is a low quality and you have to repeat? Well, do you have DNA held back to repeat? Are they going to charge you for that repeat? You should ask about their contingency plans. And then the sequencing platform, again, goes back to what you're actually looking for. Shorter, targeted sequences or longer sequences. And that can dictate the turnaround time, but also the quality of DNA, the sequencing platform. And there are at the moment, only a handful of dominant sequencing platforms in the world. There's Illumina, there's PacBio, there's Ion Torrent, there's Nanopore. These companies make sequencing machines as well as chemistries. So you need to understand, at least by name, what it is your provider is going to do.
Greg, and I think that's a very important point. There are a limited number of platforms, as you pointed out. However, some of the answers you can get are quite a bit different between those. So you need to do your homework, and hopefully we'll get to that as well throughout the course of this webinar. So one of the things that Megan asked earlier was about the kits that are used. And in terms of kits, you really need to do your trial and error. And I said, as I mentioned before, your homework. Understand what each kit offers. If you're going to be shipping these kits with these kits, you need to select the best one for your application. Don't rely on whatever is just affordable. Go out there and study these. Try shipping samples with different kits and see what the outcomes are, and you may be surprised by these. One of the studies I'm going to show you right now is to show you that all extraction DNA kits are not equivalent. And so it's worthwhile to spend time upfront doing these kits, running these kits, so that you can understand which is the most appropriate. For example, here's a sample where we have a very precious sample. It's a vaginal swab, and we want to get the most information out of this as possible. So we're going to look at 3 different kits, and we're trying to get both the microbiota, the mycobiota for the fungi, and also we want to use it for real-time PCR, and we want to use it for microbiome analysis. Next slide, please. So here's simply just running these kits, extracting the DNA on these swabs, and then seeing what's the yield and the quality of the DNA that you get out. So here simply you can see there's a couple of kits look quite similar in some aspects in terms of purity, but not necessarily on the terms of yield. Which is better? We don't necessarily know it this way, so we need to go to more detailed questions. We look at real-time PCR, for example, to see if we can't tease out this information. And I'm not saying one kit's better than another. All I'm saying is you do need to compare, because for your application, it may be considerably different. Next slide, please. So here's, if we just look at in this case, this is the 16S sequencing. So this is the total of the bacteria. Across the top, each column represents an individual. And there's 3 replicates of each individual, so you can compare the technical replicates for the individual. These are not biological replicates, these are technical replicates. Then if you look across a row, that is by kit. And again, I'm not promoting one kit over another. This is the way they are. You can see the kits are not equivalent. They give you different outcomes. Some may give you a different diversity than the others. So do you want high diversity, or are you settled for mediocre diversity, because that may be better? And the other important aspect here is make sure that you run a spike control. So you run something, that is my farthest right-hand column here in this case, so you understand what you expect to see. Next slide, please. If you look at the ITS, though, this could be quite a bit different story indeed, because here, one particular kit is extremely good at picking up the fungi, the yeast that are present in these samples, whereas the other kits, in this particular case, did not seem to allow us to extract sufficient DNA to do the analysis. In fact, it didn't even pick up the spiked sample. So something happened in this particular situation. So again, I'm not saying one kit's better than the other, but make sure that you go in and test the kits. And that's my drive-home message today. Don't expect them to be identical just because you purchased it from a company that said that. So make sure of that. Next slide, please. Oh, robotics. These are important, and Greg will talk about this as well. Certainly, you have to decide you're going to go through manual or versus high throughput. In our case, we need to go to high throughput. We've got thousands of samples coming along from our clinical studies, and we need to be able to work through these in a cost-effective fashion. So if you go to robotics, you can actually save cost on your sample analysis and save considerable time in terms of the individual who has to process those samples, allowing them to do other things like the analysis of the output of the microbiome analysis. Next slide, please. Choice of sequencing facilities. Again, going back to what Greg said, that's perfect, because selecting a sequencing platform, okay, pretty much you're going to find out that everybody has one particular sequencing platform or another, but try before you buy. Send your samples out to 3 or 4, 5, 6 facilities, whatever you think it needs to do, so that you can understand what kind of outcome you expect to get from the company. Do mock community analysis. You need to be able to understand if they're able to detect the microbes that are specifically of interest to you. Greg already mentioned this. Yes, sorry, Greg.
Dr. Tompkins, if you could just explain what a mock community is.
Okay, so we're going to go through that. We're going to see an example of that later on. But basically, it's a set of organisms that you assemble. You know the quantity that you've put in there, and you would expect to see that outcome once you do your analysis as well. So I'm going to lead you through some of the examples. And we're going to actually see, looking at using that mock community to look at which region. So the targeted amplicon region of the 16S we could look at versus another system, the PacBio system, for example, where you get the full 16S region. So spend the time to identify your target and your facility that you want to work with, your provider. Next slide, please. Okay, so here's an example of, okay, so this is just going on to the tools of assessment. So we talked about amplicons, we talked about targeting sequencing. We need to know which portion of the 16S that you want to target before you even begin doing your analysis. You can get a long way down the road and find out, oh, we're only using V4 sequence data, and we're missing a good portion of what's out there. So you need to understand what's the most appropriate target for the area you want to look at. It could be V3, V4 for probiotics, like I use, but it might be V6, V8 if you're looking for some certain pathogens. So understand what the regions are that are of interest to you, or maybe you just want to go save yourself some headaches and just go full 16S RNA sequencing through a PacBio type of system. Next slide, please. So then you have to do the assessment, and Greg's already covered this already. You have to use these databases for comparison. So you need to understand which particular database is appropriate for the level of investigation you want to do. Do you want to get down to the family level, or do you want to get down to the genus level? And is there such a thing as having too much resolution? Absolutely. You'll find out that a lot of these databases are not curated. There's a lot of garbage inside of them. So you put your material in and you come up with a whole bunch of uncultured material. But maybe one of those hits out of there is the microbe of really of interest, but it's getting lost in the background information. So you really need to think about, is that is your microbiome probiotic sequence, is it in the database? And which database should you be using? So I would test this out before you get too far down the road to make sure that it's appropriate. For us, we found that making a customized database turns out to be the best solution. Next slide, please. So here's our mock community. So here's a case where we've done 10 strains. We can do 23. We currently is our largest mock community. So you grow these microbes, you put them together in a combination you can use flow to make sure your counts are correct. You can also use in silico analysis to understand how many S16 copies of the 16S gene you want there, so you can understand what the ratios will be. Even if you make a one-to-one solution of 2 microbes, that could be very different if you looked at the in silico concentration or presence of the 16S gene. So you need to understand that and take that into account. You take these communities, you do from blends of them, you send them off for 16 applicon sequencing, or you can do the full-length sequencing to get you down to the species level versus the genus level. Next slide, please. So the output might look something like this. So here's our control strains across the top, where we've run each individual strain, and then the next ones are where we've looked at the blends of bacteria, where we've given 1 to 1 to 1, basically the first one, and then the other one is varying ratios. And what you can see is that in this particular case, the V4, V5 is not particularly looking good for the detection of the lactobacillus and bifidobacterium. They're marked in the reds, the ones that are misidentified or not even identified at all. If you look at the V3, V4, well, that's a much better situation as the V4, but the V4 reads are a little bit too short for what you might want to do. At the species level, well, we're having difficulties with the database to detect these strains as well. So we need to be able to have some flexibility, but do these types of analyses before you get too far down the road. Next slide, please. So here, so the last slide, we looked at which region, applicon sequencing region to look at. Now if we take that region, the V3, V4, which we've identified, and we look through how does that work in the different databases? How does the databases, are they able to pick up my microbe? So in the case of the V3, V4, we found a very good correlation between the Silva database in this particular situation, but the Green Genes database was still missing at least one of the microbes. The thing that we've been going to now is, okay, let's create a customized database where we can overlay our microbes into these databases, and does that improve the outcome in the databases? Sometimes yes, sometimes no. So again, be careful of that. As you can see in this particular case, it really didn't help in the situation where we put in a custom database, but in other cases we can see that it does clearly help improve the efficiency or the correctness of the reads that we're getting, of the outcomes that we're seeing. So it can adjust the composition based on that. Again, it's just composition. Doesn't tell us much about the functionality of the microbes that are there, but this is a very good place to start. Next slide, please. Here's a case where we've gone with the full length, and a case here now you really begin to see that we've lost the resolution that we had with just looking at a particular region. Why is that? There's a lot of misidentification when you get down to these lower levels. Again, database is very important when you're looking at these particular when you decide in which region you're going to look at. So again, test this out before you say, okay, well, I'm just going to do full length. That sounds the best solution for me, but you could be missing quite a bit. There's where you can have an opportunity with your custom database. There's where you can make the difference in improving the correct identification of your microbes. Next slide, please. So I would summarize by saying, don't accept everything you see in the literature or you receive from your sequence provider. Challenge them. Make sure that they can give you a good understanding of what you are seeing. Do your background work to verify the sequence provider and the choice of amplicon region. You know, I think amplicon sequencing is probably going to go the way of the dodo in a few years, but right now it is the cheapest way to get into the microbiota, and it certainly will give you quite a bit of compositional information that you don't have to worry about the other aspects that you would receive if you got a shotgun sequence data, where you're getting a lot more information and it's much more difficult to interpret, and you really need a very strong bioinformatics skills to sort through. Use mock communities to validate your choices. Do the literature search, understand what microbes that you really expect to see or would like to see, you want to evaluate, build the communities based on that, and then run these through the various pipelines that are offered by the provider. Use real-time PCR to validate the changes in the microbiota. So if your provider comes back to you and says you have this change in the composition, go back into that sample with primers specifically for that microbe, whether it's the genre or the species, and to see if that really holds throughout all the samples that you've provided. And each step must have controls and a standard operating protocol. If you don't have these controls, you don't have the standard operating protocol, then again, you're going to be misled and misinterpret your information that you could easily avoid simply by doing the background work up front. Thank you.
Okay, now we will pause for a short Q&A session, and then we'll get back to the topics with our speakers. Megan, do you see a first question for us?
Sure. So I guess a couple questions. In terms of technology, Eurofins has many different sequencing systems. Dr. Tompkins, you highlighted review of the different platforms, review of the different kits. At what point would you consider changing to a different platform or changing to a different kit, understanding that you as a company may have history with one platform, one kit?
Exactly. I think one of the things that why we originally brought everything back in-house to do the analysis is because we did see that every investigator that we were working with, every CRO that we were working with, were using a different provider, and the outcomes were absolutely incomparable. So one of the things that we decided to do was to standardize it. We're not saying that we're doing exactly the best way, but we are doing in a consistent fashion. We're making consistent errors, which we are quite aware of. So we are using, you know, we found our provider that we can work with. As Greg pointed out, this is very important that we can work with that individual to understand what we expect to achieve. And that's, I think, probably more important necessarily than some of the compositional outcomes that you would see. Make sure you do the best job you can of it, but having somebody who can appreciate what you're trying to achieve, I think that's the critical aspect as well. So a lot of the outcomes will be based on your ability to interpret, and I think that's an important factor that we shouldn't forget.
Thank you. Thank you. Now let's continue on with the next question.
Okay. So before we move on to our next question, Dr. Siragusa, did you have anything to add to Dr. Tompkins' point?
Yes, I did, and that is to distinguish mock communities or standards that you might customize into a database or use to make a customized database. In the case of fecal samples, you might or might not know what to expect, and virtually 600 plus different species in feces, you won't really have a great idea. It's an unknown. However, if you're a manufacturer and you're asking a question about your mixture of strains, Those are two different things. So one can be a mock community that better reflects fecal communities, and the other can be a mock community or a customized database that contains your genus and species of interest.
Yeah. And Greg, I think that's a very important aspect. One of the reasons we first got into microbiome analysis was that one of the, from a regulatory perspective, one of the governments came to us and said, they looked at our sample, they were using 16S V4 regions, and they said, oh, well, you know, your product doesn't have the strains that it's listed. And I really had to laugh because, you know, you cannot use this to get down to the strain level. And, you know, again, they were trying to use a database that was built for fecal analysis and trying to apply it to microbes that are very defined. And so we did, this is where we got into these community analysis, and we clearly could see that, that, you know, these communities were just not picking up our strains. So that's why we started to build these back into the databases. So make sure you're quite right, Greg. Make sure that your microbes are also part of that database when you go to look at it.
Great, thank you. So that's actually also a really nice transition in terms of understanding the limitations of microbiomes, the level of transparency or depth that a microbiome analysis can achieve. So Dr. Siragusa, could you speak to some of the limitations or additional analyses that you would consider when performing microbiomes?
In terms of additional assays, you might benefit by doing a culture-based microbial profile. If you have a mixture, now I'm talking about products, or probiotic products. If you have a mixture and you can separate out things based upon culture, probably not a bad idea to have that information in hand. If you have quantitative real-time PCR assays for specific targets, be they genus, species, or even strains, that's an additional assay that you might want to use. And then Dr. Tompkins is going to talk a little bit more about metabolomic analysis in great detail, but I mentioned earlier end product meaning what comes out the other end of the animal in this case, and might be volatile fatty acids. It could be bile markers. It could be a number of things that are single metabolites or multiple metabolites, which is the metabolome. All of that goes into that big pile of data that we call metadata. And the metadata can be geography, age, gender, ethnicity, race, etc., or it can be chemistry. It can be levels of volatile fatty acids, short chain or branch chain. Limitations. Currently, most microbiome procedures are not a rapid method. Actually, the technology is moving toward approaching about the same length of time that it takes to do a lactic acid bacterial count, and it's 3 to 4 days. It's approaching that. But rapid response is not a descriptor for microbiome analysis. It takes time. I mentioned earlier, this is not a precise analysis. It's a proportion, so we're comparing proportions. And there are very specific statistics for comparing proportions versus linear or continuous variables. Microbiome analysis detects DNA. Now, whether that DNA came from a cell that's viable or not is not reflected in the microbiome. Therefore, you might want to augment that limitation with qPCR or even culture. And then I would add that databases change nearly constantly. And so for your purposes, if you use a customized database, you have to make sure that it's up to date. And by you, I mean you and your provider. If you use public databases, just be aware that taxonomy can change. So names can change, but it's the same gene sequence. Next slide, please.
Exactly. I think, Greg, you brought up some excellent points there. What we've done to, so, even though we're using the microbiome compositional analysis, we need to be able to still understand whether or not we can still identify our microbe in that matrix, whether it's a fecal matrix, a vaginal swab, saliva, whether it's breast milk, for example. So we've done whole genome sequencing of our microbes. We found the unique genes within those microbes that allow us to develop real-time qPCR primers for that microbe, and then we can apply that to the sample, any sample that you so desire, including the finished product. So these are ways of being able to detect your microbe specifically in amongst everything else that could be present. There are other techniques you can use in digital drop PCR now as well. But you have to be aware that there's certain occasions when ddPCR may be better or worse than the real-time PCR. For example, I can use ddPCR very well for looking at product analysis, but certainly I can't apply that as easily to fecal samples at this time because it's just not, we're just not able to get the ranges sorted out very well. So there's opportunities there and things that can be worked on and maybe other solutions there as well. In terms of viability versus survival, we use flow cytometry. So just ensuring that the DNA doesn't ensure anything about the viability of that microbe. So you do need to use other techniques like flow cytometry. And then we need to get away from strictly compositional analysis, and we need to look into functional aspects of the microbiome. So you can use things like looking at the metabolites, such as short-chain fatty acids or branched-chain fatty acids, or you can look at other aspects. You can use the fecal water, for example, to look at host immune markers or microRNAs. There's many things that we could look at in terms of the compositional changes in respect to changes in the microbiome. And then in vitro modeling, how do we design a model that will allow us to look at some of these aspects, the biomarkers or the functional analysis that we want to look at, and how can we use those models to predict what will happen in a clinical trial, and thereby maybe engineer our trial a little bit better to look at those groups that may respond better in certain situations. So that's a whole other area of conversation as well. Next slide, please.
Thank you. So it sounds like there's a wide range of additional assays and benefits to using microbiomes in what we're doing today. How do we move beyond what we're currently doing? What are some of the new applications that you see with each of your customer base? Dr. Siragusa.
Okay. The first that came to mind is actually, again, using microbiomes to understand the baseline of animals in your studies. If you're doing animal models, most of what Dr. Tompkins talked about were human trials, and so you can do the same thing with animal models, be they mice, rats, or whatever your model is, to figure out a baseline. You can ask for customized databases that reflect your specific strains. I see that people are moving more and more towards that. So the public databases might not necessarily be appropriate. A little bit longer term, I think we're going to see faster turnaround times for microbiome analysis. And in the same time period, I think we're going to see a shift from targeted microbiomes, which we discussed a lot today, to shotgun metagenomes, which is a technology that's up and coming. There's some very good providers of it today. It's just not as common as targeted microbiomes. Those are the major points that I see coming down the pike. Next slide, please.
Thank you. And what about you, Dr. Tompkins?
Well, yes, I think where we get at today, well, we're beginning to utilize or looking at how we can utilize the microbiome composition to screen participants for inclusion and exclusion trials. We understand now that there are certain responders, compositions which differ from non-responders, and we need to understand how do we incorporate this and stratify these participants in our study, potentially based on pre-screening of their microbiome. So I hypothesize that one particular group are going to respond because they have a certain composition more so than another group. For example, let's say I have a low bifido group and I want to see if I can increase their bifido group. I might select a population that has low bifido to begin with. But you need to understand the range of your participants prior to the big onset of the trial. That would be one example. We also need to get to functionality. One of the areas that I like to look at is the resistome. So the totality of all the antibiotic resistance genes. And you really begin to see that these are functional markers of the activity and potentially can correlate again with certain conditions. So we see, for example, individuals who have gone through a lot of antibiotic therapy treatments will have a very high load of antibiotic resistance genes, whereas people that we use for fecal microbial transplant donors, they tend to be extremely low. In fact, almost non-existent in terms of their antimicrobial resistance genes. So that's obviously something that you want to be able to evaluate. And the other area is taking us into new areas, microRNAs. I love these as a target. I think this is possibly a means of host microcommunication. It's maybe the basis of some of the crosstalk that we see, where these microRNAs can be coming from the host, and we have a good understanding. We have some understanding of how these microRNAs coming from the host can be influencing different systems, but we know also that the microbes themselves are capable of producing some small microRNAs or small RNAs or microRNAs, as we may call them. And how are these influencing the host as well? And I think that's another step that we need to think about in the very near future, is using these as potential biomarkers to see again who may be responding and who may not respond in certain situations. And then there are many, many other areas to consider. We talked about looking at the microbiome, so the yeast and fungi are present. A lot of people talk about the virome and the viruses and the phage and the protozoa as well. I mean, and within that, they have their own microbiome. So there's many levels of complication. You can just keep on digging in this mess. It's quite fun, actually. So it's been a fun ride, and we have no limit. But we need to get away from looking strictly at compositional analysis and begin to look at functional analysis, because there's a lot of redundancy within the microbes and their ability to perform actions that are independent of the genus of the species. So I think that's where I was, again, a word of caution.
All right, well, that completes the presentation portion of this webinar. We have about 3 minutes, so let's look for some attendee questions. During this time, you can continue to submit questions. Any questions that go unanswered during the Q&A session will be followed up with by our hosts and presenters. Okay, for the first question, there are some chloroplasts from plants that will cross-react with primers. What do you do in those instances?
I'll take that, Dr. Tompkins, if I could. We're taught in microbiology that chloroplasts and mitochondria might actually be bacteria that were swallowed by other cells. Well, it turns out that mitochondria and chloroplasts have some sequences that will be amplified by 16F primers. And one way to avoid that is by choice of a different primer set, but probably an even better way is to bioinformatically subtract out those chloroplasts and mitochondria, because the database, if it's good, will say these are not bacterial, even though the sequence is similar. So it can be removed in silico.
All right. Thank you, Greg. And I think we have time for one last question, and that question is, how do we decide how many samples to have in a treatment group to have a statistically significant comparison or correlations, understanding that there is a variation?
Right. I'll start with this, Greg, and you feel if you want to jump in. So from our side, I mean, when we're designing a clinical trial, we don't based usually on microbiome composition. We're usually powering our studies on other clinical outcomes that we wish to see, and we do the microbiome as a backup to that. That being said, sometimes you want to look specifically at, say the changes in lactobacilli in the bifidobacterium. So you try to power based on the evidences there. However, the variation in human individuals is quite large, and so getting a representative calculation or a power calculation in those particular cases is very difficult. Some people like to use quite small levels. I don't think that's appropriate. I think you need larger cohort studies to be able to assemble this information. And Greg, maybe you've got some different experiences from other areas as well.
Sure. Very quickly, talking about animal models, for instance, an avian model, if you're doing a nutritional trial with broiler chickens that you're feeding a probiotic, you might have, within a treatment, you might have 100 birds or more. In a house, you might have 20,000 birds. Well, right now, we're not going to do 20,000 microbiomes. But for statistical significance, even though you know something about the baseline of those chickens and you know what they're fed, you have a good opportunity by microbiome analyzing all of those birds in a treatment group or a subset. So we're talking about dozens to hundreds.
Yeah, that's the advantage of working with animals. They have a lot less variability, I should say, in their microbiome. So it's very nice that you can do that, and they don't mind giving samples.
and I guess you don't feed humans lab chow.
No, we have to look at that as well. What do we feed them? Correct.
Great. Well, thank you. That wraps up our Q&A session and the webinar. I want to remind everyone that a recording of this webinar, along with a copy of the slides, will be available by the end of this week. Thank you, Dr. Greg Siragusa and Dr. Thomas Tompkins and Megan Spike for their expertise and their presentations. Thank you, everyone, for attending.
Thank you.
Thank you.
Bye-bye.
Is anyone else still online?